Hi, my name is Tim Downing. I work at Dublin City University. I'm a lecturer in genomics, and today I'm going to talk to you about the population genomics and genomic epidemiology of Leishmania parasites. This is a recorded lecture, and therefore I would ask that you tweet me questions in relation to this topic, perhaps using the hashtag #eponge, and you can tweet me to my Twitter address here and also the #eponge Twitter account here as well. So what I'm going to discuss is a basic introduction into the population genetics of Leishmania parasites. Some of this may be new to you, some of it may not. So feel free to tweet me any questions on this to this account, and I will answer them. What is useful is that we want to create a community of people working on Leishmania population genetics, and therefore you can chat to one another about your queries and your answers and your thoughts on different topics. And because we all have different backgrounds in this general area, I'm sure we can have many different opinions on how best to go about maybe designing projects and thinking about Leishmania genomics. So I'm always looking for new people who are enthusiastic about Leishmania genomics. There are many different things that we've worked on. I worked not just on Leishmania parasites, but also on gram-negative bacteria like Escherichia coli, gram-positive like staphylococci, and other species in the escape set. What I'm particularly interested in is their population genetics, population structure, their evolutionary genetics, the adaptation, the origin, division, transmission, and spread of different strains, particularly drug-resistant strains of any species, so that we can study the pangenomics of different strains, and also if you're interested in the functional genomics, that can be explored too. So this is my lab website, so you can read more about it here. You'll also have the slides from Chris, so that feel free to look at that in your own time. So what I'm going to talk about today is the general background of the Leishmania genus and the genomes that we have available to work with. I'm going to give some background to Leishmania population genetics, some of which you may know, some of which you may not. What I'm going to focus on in the interest of time is this study by Franza and al. Recently preprinted on the bioRxiv just a few months ago on the Leishmania donovani complex. This is to give you the state-of-the-art knowledge on current research in this area. What's really great about the study is that it collects isolates from around the world, 151, as shown by this diagram here, and really gives us a cutting-edge information on the whole area of Leishmania donovani complex genetics. So, first, I'm just going to give you some background on Leishmania genetics and the genomes. So, as you're aware, with this neglected tropical disease, it affects many countries around the world, particularly the subtropical and tropical regions. There are at least 20 species, many of which are really understudied, not well tracked, and not well understood. Many phlebotomy sandfly vector species are able to transmit this parasite from person to person. So there's a digenic life cycle where it goes from human to parasite, human to vector, human to vector. And there's a large number of sandfly vectors that are associated with this. An additional consideration is the diversity of pathologies that is perhaps associated with the host immune response as well. So in humans, we tend to see most commonly the cutaneous forms affecting mainly the skin, forming lesions there. It's typically mild, and that's associated with the majority of the global disease burden. Less frequently, we see the visceral form, which is much more acute, often associated with death, where the leishmaniasis spreads to the internal organs. This is much more severe, and it certainly is a major concern. Also, more commonly in the Americas, we see mucocutaneous leishmaniasis. This is a moderately severe form of the disease, particularly affecting the face and particularly the nose and sinuses. Another feature of the disease is post-Kala-Azar dermal leishmaniasis, which is essentially a relapse following low levels of parasitemia associated with the cure of the patient. We see a subsequent relapse, which may occur a long time after the initial case. When you put these all together, there's an estimation of 1 million to 1.5 million or so cases annually, and these are commonly associated with people who are immunocompromised. They're complex cases, complex diseases. There's a major socioeconomic considerations in terms of the poverty that is essentially associated with disease and at outbreaks of conflicts around the world, such as those associated, for example, with the Syrian crisis, where people are not given appropriate healthcare and therefore they are much more prone to having these diseases. So if you're more interested in the background on this, I urge you to go to the Gradoni, his book chapter, her book chapter on and the introduction to leishmaniasis, recently published. I highly recommend this. Another feature of Leishmania is the life cycle. So with the human stage on the right and the sandfly stage on the left. If we start off here where sandfly bites a human, it will inject promasticate parasites into the human. These will be phagocytized by macrophages. This may result in the parasites being eliminated, but sometimes they may be able to survive intracellularly inside the macrophages, grow, divide, transform into the amastigote stage, shown down here, from promastigotes. So it's important to note that promastigotes and amastigotes are metabolically, biochemically quite different life cycle stages, and therefore that's a major consideration for studying Leishmania parasites. Following this, we may have a sandfly that takes up parasites from this infected person. Inside the sandfly, the parasites will progress through the sections of the gut of the sandfly over about 7 to 14 days. They'll transform from amastigotes into post-cyclic promastigotes. They will undergo meiosis at this stage and eventually become metacyclic promastigotes that are available to be injected into the human host by a sandfly when it bites the person. This is quite an interesting area which deserves ongoing research. So when the promastigote secretory gel that forces the sandfly to mix the parasites that are released into the human bloodstream, we know that not all of the promastigotes are injected into the human host. So some metacyclic promastigotes can actually de-differentiate back when the sandflies have a second feeding. So they can undergo a sort of a reverse metacyclogenesis at this stage, and this is important because these parasites, if the sandfly has a second feeding we see much higher virulence in these parasites. So these older retained metacyclic promastigotes remain inside the sandfly, and in some way or other are associated with much higher virulence in the subsequently injected parasites. So the retention of these Leishmania parasites, the parasitemia levels, is much higher, and this is shown by a paper in Nature Microbiology last year. And what I recommend is that you look at this paper on revising Leishmania's life cycle and the associated paper to get a better feel for the complexities of Leishmania life cycle. So these retileptonoma promastigotes appear to be associated with much more severe infections in humans. So this is another feature of Leishmania, is the two-stage life cycle. So let's look at the overview of the history of Leishmania genetics. So you can see more about this in a paper by Cruz and Freitas Castro on genome and transcriptome analysis of Leishmania species down here. So taken from this project, we see that the Leishmania Genome Project was launched in about 1994, which culminated in the Leishmania genome being finished in about 2004. In the following year, the comparative genomics of Leishmania major, Leishmania infantum, and Leishmania brasiliensis was published. So this is really a step change in the whole area of Leishmania genetics. So moving from very basic biomarker testing to much more advanced work. This was followed up by better databases to enable Leishmaniacs to study the parasites much more effectively, and also more advances beyond genomics, looking at RNA-seq data and other types of multi-omic data, so that more incisive functional genetics testing could be implemented. More recently, extensive work on field isolates has been completed, and also long-reach technologies, PacBio, Illumina, Nanopore, and BioNano, and so on, have enabled much better reference assemblies to be developed. And this is important because Leishmania has a number of genomic characteristics that are quite unique, and therefore we need very high accurate reference genomes in order to be able to study it properly. So when we think of Leishmania as a genus, it's worth considering the diversity inside it, at least in terms of its taxonomy as a way of navigating it initially. So what you see on your left is the subgenera. We have the subgenus Leishmania, the subgenus Leishmania Leishmania that mainly infects lizards, as opposed to Leishmania that infect mammals, Leishmania viania that mainly infect human and animals as well, and then other assorted subgenera that are less well documented because they have a lower generally global burden of infection. Within each subgenus, we see that there are different complexes. In the Leishmania complex, there's 4 main species complex, which contains a compendium of subspecies within each one. We see the main species complex that is known inside the Leishmania is Tarantulae, and the main one in Viania is Brasiliensis. There's a range of subspecies inside here. Some people call them species. The level of differentiating here is much lower compared to that between complexes, compared to that between subgenera. We know this because if we compare the different species here, we can identify different levels of complexity across these different species. So if you want to read more about this, I recommend the Kundi et al. Paper that reviewed this whole area from 2 years ago, and they looked at the Leishmania subgenus versus the Viania versus the Leishmania, and also other more recently documented ones like the Mundinia, including Enreti martinicus and other ones. What you see here, there's other less studied Leishmania like Paraleishmania, and as well as other species in red here that are Leishmania species of interest, but maybe in a very restricted geographic region. So we're going to context on the Leishmania Donovani complex later. So I was involved in some work led by Simone Coughlin in 2007, where we looked at diversity across a handful of housekeeping genes in the Leishmania on your right-hand side and the Leishmania on your left-hand side. So what we see if we compare 2 subgenus is a large genetic distance between them, and much less genetic distance within each species complex and across the subgenus here. So we know that subgenera are quite different, and we can zoom in then on the Leishmania Donovani complex, compare that to the Leishmania major complex, including Arabica here, and then the Tropica complex shown here. So we have this level of diversity within Leishmania subgenus. Not all Leishmania species are represented by genomic data. Leishmania major, Brasiliensis, Donovani, and other complexes are very well documented, but some are not. So it's worth considering that if you're doing any studies, that we have many reference genomes, and therefore, if you're asking the question, what reference best to use, it's a really, really important question. Not all references are the same, and you perfectly want to use one that is representative of your sample collection to avoid biases associated with extreme genetic distances. So I just point out that there are a range of references, and you may want to use not just one, but multiple ones if you want to study your collection properly. If we're looking at the Leishmania Donovani complex, we mainly use this reference here from Spain in 1998, known as JPACM5. This was our main reference for that study. I'll talk about later by Franson et al. So, in terms of Leishmania population genetics, moving on from the genus and the genomes, there's a number of questions we can ask ourselves. Firstly, and this is something we'll look at later, the population structure. Do we have panmixia? How much of a role does geography play in the genetic diversity that we see? Migration over time. Do the parasites spread widely across geographic regions, or are they very restricted? We know that Leishmania can undergo meiotic divisions inside the sandfly, and therefore we can see mixing cross-attenuation and therefore recombination, but that requires parents that are quite divergent in order to be able to detect it. So how much mixing do we see? Another consideration and a feature of Leishmania is aneuploidy. So this is differences in the chromosome copy number in a given cell. So rather than having 2 copies of each chromosome, sometimes we find that 3 or 4 copies of a particular chromosome, and this is very fluid. So even if we have 2 genetically very, very closely related samples, what we might find is that one has 3 copies of a chromosome, one has 2 copies of a chromosome, and it's an intrinsic feature of Leishmania that have universal aneuploidy. The chromosome copy number change extensively. Further considerations are: what question do you want to ask? What scale are you looking at? Are you looking at diversity between the species complexes, maybe within a subgenus? Are you looking between different species inside a complex? Are you looking within a species that is spread around the world, or maybe within a population that is restricted to a geographic region? Furthermore, you may be looking at a particular outbreak that is very recent in a town or district on a very local level. And then even further, we know that the parasites exist inside a host, and therefore we really want to understand better, do the parasites evolve inside the patient, or do they evolve inside the sandfly vector? What's going on there? Not much is known, and it's important to think about what question do you want to answer when designing a study. Another question is what genetic data is available. So historically, we had enzyme electrophoresis, microsatellites, and improved resolution provided by multi-locus sequence typing, so looking at a few housekeeping genes to get a decent genetic picture. MLSD can provide this, less so with microsatellites and MLEE. SNP genotyping is better, where we look at the spectrum of mutations on a chip, representative of the whole genomic data. This is somewhat biased depending on what SNPs we look at. So if we want to look at mutations right across the genome, to look at the aneuploidy, the chromosome copy number changes, copy number variants, as well as SNPs, genome sequencing is the best way to go. And this gives us a much more improved resolution because many biomarkers are essential. So in terms of designing studies, I recommend reading this paper by Rougeron et al. On Leishmania PopGen. So considerations are important for Leishmania. Does it obey Hardy-Weinberg equilibrium? Clearly, it's somewhat complex, so we have to ask ourselves that question, particularly for a sample collection that we're looking at. What we'll see is that sometimes we might have populations where we have many independent loci, be they SNPs or genes or whatever you're looking at, but sometimes we see very high linkage disequilibrium where large segments of chromosomes have a single phylogenetic signal. We also see very wide levels of homozygosity in certain isolates, but also certain isolates also have very high heterozygosity. So a lot of mutational differences between the different chromosomes inside a particular sample. So although we know that Leishmania undergo meiotic divisions, often it's not as clear if there's some sort of selfing going on here, or maybe the parents can often be very closely related. This can sometimes give rise to a clonal picture because if the parents are identical, we can't necessarily identify recombination signals for that particular the progeny. However, we do know that there are many inter- and intra-species hybrids in Leishmania that have genes for meiosis in their genomes, and since 1990 we know that they undergo nuclear fusion, as shown by Lanaut and Ryu. So what we want for our study is that we have used genome sequencing ideally to give us a sufficient density of biomarkers to really say a lot about the gene functions, but also the population structure of our sample collection, and we need really broad geographic sample to get a better sense of what's going on. Further things to consider are the sample collection design and the question that you can actually answer with your sample collection. Sample preparation is important. Ideally, we'd take the sample, the DNA, direct from the isolate, maybe using probes or other approaches to directly tackle the original DNA, rather than current methods which often culture parasites to create a clone of parasites whose DNA can be extracted and then PCR. This introduces biases. We also need to consider that if we're doing, say, genome-based amplification or other PCR-based amplification of DNA, that there may be plasmid-specific biases which should be considered. Another feature is, can we infer the haplotypes, the SNP co-inheritance patterns across chromosomes? And also we need to consider that if we're looking at genomic sequence reads, how long are they, or how long is our PCR product, and can that actually give us information on the wider segments adjacent to that particular portion of interest. So, as I mentioned, aneuploidy is a major feature of Leishmania associated with the extra chromosomes. In short, it's associated with some sort of meiotic mistakes in prophase 1 of meiosis. So we should have tetrads here, as shown by the cell down here, that line up in the middle of the cell. We have the chiasmata joining the tetrads together, and we expect that two chromosomes should segregate and be pulled towards the poles of the cells away from each other. But that's not the case for Leishmania. Sometimes we get some sort of 1:3 ratio instead of the 2:2 ratio that we should expect. So there's a number of different hypotheses in terms of what components are defective, and multiple mechanisms may be possible. In terms of the genomics of this analysis, well, due to these mesomeiosis, we see that a small minority of chromosomes become trisomic, and indeed we know that chromosome 31 is typically tetrasomic. So there's 4 copies of chromosome 31 nearly always. Sometimes 5, again, due to the same defective meiosis. But it's worth noting that most chromosomes are disomic, and therefore we can apply aspects of population genetic invariance to those disomic chromosomes. Another item that was studied by Coughlin et al. Was in the short Leishmania complex. We found that there was a different number of chromosomes than the default 36. So this is another feature of Leishmania, that there's chromosome fusion and chromosome fission. We might have extra chromosomes or fewer chromosomes. So it's important to think, broadly, maybe there's something different about a particular set of samples. In this case, what we found in this sample called Leishmania adleri HO174 was that its coverage switched right at this point in the chromosome. So this is chromosome 36. We saw that its coverage switched here from being about disomic compared to the other chromosomes to being trisomic up here. When we looked at this breakpoint, we were able to use Leishmania major origin of our DNA replication points to show that this actually coincided with that point here. And if we separate the two chromosomes out, what we can see is that it is likely that one is disomic and one is trisomic, and therefore they are 2 different chromosomes rather than being 1 chromosome. Similarly, in a different Leishmania adleri isolate, we found evidence of a higher chromosome copy number for this portion here and lower for this portion down here. So the same principles will apply, and these are 2 different related isolates that have different numbers of chromosomes. So this is worth considering into your Leishmania studies, to probe, ask very, very basic questions about your Leishmania parasites, because they may not obey all the basic principles that we typically assume. So, in terms of the population genetics, it's important to just recap on the process of looking at different types of SNPs inside Leishmania. So if we have 2 parents, represented by a green and blue chromosome here, a yellow and black chromosome here, we have the progeny here shown by a green and black chromosome. If we get multiple rounds of inbreeding among these progeny, what we'll see is a mixture of progeny haplotypes at the end. What this will result in is that initially we had progeny that were uniformly heterozygous up here, but after multiple rounds of breeding between the progeny, what we'll find is some segments down here for this pair of chromosomes in a single sample is that some parts are black, some parts are green only, shown down here as green and black, and so some parts are heterozygous, shown by the red parts, so they're green and black. This means when we're looking at hybrids in Leishmania, you have to expect that some parts, if there is extensive inbreeding, might be homozygous for one of the parental alleles, and other parts may be heterozygous, they have a mix of the both. And this is what we've seen in previous studies that have looked at these particular set of samples from Kukurova, in Turkey, where we saw across a set of 12 samples certain regions that have high density of SNPs, shown by these coloured bars here, and certain regions that have far fewer SNPs. So what we did with these samples is we mapped the reads against the reference chromosome for Leishmania and Phantom. We saw some regions like this white block here, where we saw no SNPs because its DNA had an exact agreement with the reference genome. And so you have other regions like the colours bar here shown for Cook12 that has a very high density of heterozygous SNPs. So this region appeared to be different. So what we saw was that there are some regions that are like one parental stain shown in black, other ones that are shown like the JPCA5 L-infantum reference shown here in white, and some that are mixed in red. And what you see here for this set of very closely related parasites is that the segments differ. Furthermore, the segments have a kind of block-like structure. So this is similar to our model shown here. So you see blocks that switch from one ancestry to another ancestry, or to somewhere in between. What is interesting about Leishmania, because of aneuploidy, is that we can actually look at the across a particular chromosome, shown on the y-axis here for chromosome 4, we look at the read allele frequencies. So we're looking at the number of DNA reads as a percentage as they map to the chromosome, and we look at the number of reads that are different or similar to the reference, going from 0 to 100% here. What we see is that sometimes we get a lot of reads at a particular place have a 33% match with the reference chromosome, and other ones shown in blue here where they have a 66% match with the chromosome. And what we see at about 125 kb to 150 kb is that they match a trisomic pattern. So if they're disomic, they'd be 50-50 in the middle, because 50% of the reads would have one allele, 50% of the reads would have another allele, but here we see it's a 1/3, 2/3 ratio. And sometimes you can see from 260 kb onwards, they switch. So you go from green at 1/3 to green at 2/3, and vice versa for blue, Cook4 across here. So if we put together the different features in Leishmania, we actually make better inferences about its population genetics. And this study showed that there was mixing between these different isolates from Cucurova, and this kind of formed a basis for looking further at the Leishmania-Donovani complex, particularly because these samples, Cook, as they're called, had a mixed ancestry, similar to Leishmania infantum, but somewhat like a different unknown ancestor that at the time in 2014 we had not sampled. So let's just recap on this. If we have diverse parents, like one parent here that's green and blue, another parent here that's yellow and blue, and they hybridize to form progeny, we'll have a sampled recombinant progeny that has a mixture of ancestries, some of which are like parent 1 in green, some of which are like parent 2 in black, and some of which are mixed. This is distinct from a separate lineage. So what we'll see for a hybrid lineage is few new SNPs, but many heterozygous SNPs, because we have two parental alleles here that are observed already in the population. In a separate lineage, what we see is many new SNPs and fewer heterozygous SNPs. So in our hybrid, we also see new haplotypes from known ones, whereas our separate lineage will see entirely new haplotypes. So this is different. So a rare lineage will have new SNPs, few heterozygous SNPs, and some new haplotypes. So this is a model of the ancestry here. So this is if the parents are diverse, which makes it much more easy to capture these signals. If the parents are somewhat homogeneous, we may have a different story where the parental alleles are very, very similar, and therefore distinguishing between the patterns of our hybrid and one that is rare is more difficult, because it depends on how divergent the parent was and how separate this particular lineage was as well. So what we really require here for better inference is diverse parents and diverse Leishmania. And that's what we have inside the Leishmania-Donovani complex. So that's what we're going to talk about next, the Leishmania-Donovani complex. And this sampled 151 isolates from right around the world. So prior to this, we had a paper in eLife in 2015, or submitted in 2015, where we looked at diversity between infantum here, represented by an isolate in Spain. This is mainly around Europe and the Mediterranean, or so we thought at the time. We had LV9, which is the best documented isolate from East Africa. So there's a lot of Leishmania from Sudan, Ethiopia, Kenya. That's another focus of Leishmania infection. And this is different to the Leishmania in the Indian subcontinent, represented by a sample here that represents one Leishmania lineage from the ISC Indian subcontinent, and then we have another set here that represents a second ISC2 lineage from the Nepal uplands. So the new study by Franson gives us much better information on diversity of Leishmania across the world. So what's nice about the study is that the information is uploaded to microreact, and we'll look at that later. So you can look at the frequency of different samples from around the world, across from China into Uzbekistan, into France, and so on. And what we've done is reconstructed the evolutionary history of these samples to show the differences in the genetic groups that we see. So let's go to this website, have a look around. You can do this in your own time afterwards. And what you see here, if you look at this website, is that it has a tutorial and so on at the start. But if you click on a particular country, you can see the number of isolates that are associated with this country and when they were sampled. So you can see that more of our samples were sampled in recent dates. Some, in fact, go back to 1933. That's one from Malta, a bit of an unusual sample there. And then it goes up to 2012, a sample from Leishmania phantom from the Middle East. If we look in Sri Lanka, we had a few samples there, quite a few samples from China, and also samples from of Leishmania phantom from Brazil, parts of Central America. So the 151 don't include all the Leishmania that were available, but they're representative of the diverse types that we see around the world. If we go back to the Turkish kook samples, we see in green here that these are shown by this geographic region, and that's important to note. We also see in grey here more related strains, CH, that are isolated from Cyprus, and they're also an interesting set of parasites. So you can see their genetic groups here, and I suggest you have a look around microreact and other data sets on microreact to get a feel on why it's a really, really excellent interface for looking at Leishmania parasites. So these 151 isolates, most of which were cultured from pomastigotes, they are mainly sequenced on Illumina HiSeq. There's extensive work on getting optimal DNA libraries, sequencing them properly, doing proper QC, doing proper screening to identify mutations, to look at the aneuploidy, the SOMI, SNP, and CNVs that are present, and we were able to look at about 88% of the genome. Not all was evaluated because of the diversity of repeats that are present in the genome, and also the need for longer reads to really understand all aspects of the genome. So we were able to look at about 32 megabases of sequence out of about 36 megs in total. So 395,000 SNPs were found right across the genome. So a sizable number of bases were different. And we also see that if you put together SOMI changes and copy number variants, that the vast majority of genes had at least one isotype with a dosage change, over 90%. We also found inside the Leishmania-Donovani complex that there are 7 main genetic groups, and these are represented by the colours. So you can see there in blue, Leishmania infantum, the classical Leishmania infantum, is spread right around the world. This was unexpected. Historically, it was thought to be mainly in the Mediterranean and Europe, but you see that there are samples from diverse places like Uzbekistan and China, showing that it's not geographically restricted. You also see this group in red here, which is from the Indian subcontinent, which mainly occupies that region. The purple lineage is from the Indian subcontinent as well, but is restricted to really the uplands of Nepal. In wine, there's a Leishmania from Sudan and North Ethiopia, which is similar to the ones in light pink here from southern Ethiopia and Kenya. There's also a distinct lineage here in orange from the Middle East. And then there's also other Donovani shown in red that are not clearly assigned to any genetic group. Along with those, we also see there's a big, the Turkish strains from Kukurova in green, which are hybrids, and other Leishmania shown in grey that we'll discuss further, as well as a few in black who, again, could not be clearly assigned to a genetic group. So when we represent kind of our phylogeny of the different Leishmania parasites, you can see that here that we have many samples from the Indian subcontinent shown in red, and then the different genetic groups around the world have different degrees of similarity. So as you can see, there are many samples from Leishmania infantum, and some of the groups are represented by fewer samples. So you can look at this further inside the paper. What's important to note is that we see that the deepest phylogenetic branches go back to Leishmania donovani in East Africa, so Sudan, Ethiopia, and Kenya. suggesting that that might be a hypothetical origin, geographic origin for where the most recent common ancestor exists for this collection. So the resolution using genomic data is suggestive of that, and further research is really essential to clarify that, because due to the SNP density across the complex, this is a kind of nuanced question to understand fully. In terms of the number of SNPs per group, you can see that the Kenya and Ethiopia groups are the most diverse, followed by the grey group, represented by strains from Cyprus, which are associated with hybrids. They have a lot of SNPs that are unique to this group, perhaps because they're geographically somewhat restricted. And what you can see in the other groups is that they have a large number of SNPs that are restricted to that group, suggesting some genetic drift. In terms of the aneuploidy, you can see chromosome-specific variability. So there's a lot more on this in the paper. What we see is that over on the right-hand side, that there's a lot of diversity in chromosome 23, where the standard deviation of somes of the 4 largest genetic groups here was very high. So this is a high standard deviation in terms of its chromosome copy number. It is a very variable chromosome copy number. So it's typically 3, but sometimes is 2 or higher. So that's chromosome 23. Chromosome 31 here is typically tetrasomic and has a moderate level of variation from that to trisomic or pentasomic. What you see on the other extreme is chromosomes 19, 27, 28, and 34, which are nearly always disomic. So why we see the spectrum of dosage changes is not entirely clear. We know that chromosome 23 harbors a set of drug resistance genes and therefore may vary more for that reason, but explanations for this pattern across the genome are still ongoing, and there's more detail on this in Franzens et al.'s paper. Genomic heterozygosity is also of great interest. So here we see the heterozygosity of the SNPs plotted on this axis, going from 0.0 to up to 0.06. And then the fraction of reference cells compared to the Leishmania infantum genome is shown here. So these samples in blue here are very similar to the reference genome shown by the value of 1, and these samples down here, like SUCR2 and BUM3, are somewhat different. So if we look at the homozygosity axis here, the x-axis, we see that these are similar to Leishmania infantum, or are labeled blue as in Leishmania infantum. But if we look on this axis here, what we see is there are certain samples like MAM and EP that are very, very high heterozygosity. So our explanations for these particular samples are somewhat nuanced, and you can read more about that in the paper. What is worth focusing on as an example is the CH34 sample, which is potentially hybrid, and the same for this one, CH32. So they have a different level of homozygous SNPs compared to the Leishmania infantum reference, as do these green Cook samples from Turkey, which we talked about before. However, their heterozygosity levels are somewhat different. So the dotted line here indicates the level of heterozygous differences between these samples. So if you zoom in, you can see that these samples are somewhat different, and somewhat different again to these other Cypriot samples down here. So let's explore that in more detail. What is relevant here is that if you look at these samples, H32, which is a hybrid, CH34, which is a hybrid, if we compare that to the ones that are not hybrid, CH33 and 36, work by Smirnis and Guzelu et al., that was detailed more by Bossati et al. In Bio last year, shows that in fact the hybrids here were less virulent than the ones who were not hybrids. Recent work on isolates that have come from more recent parts of the Middle East and elsewhere has shown that hybrids can sometimes actually have higher virulence. So the hybrid nature of parasites is important, not because it makes them more virulent, it's because it changes their virulence, and that can be higher or lower, but it's really important to think about potential outbreaks that might be associated with diverse Leishmania that we have not really profiled genetically before. If we look at CH34, there's a plot here showing the different chromosomes going from chromosome 1, 2, 3, 4, 5, and so on around our clock to chromosome 36. This outer window shows heterozygosity at about going from 0 up to 98 SNPs per window. What you can see is that some regions are very homozygous, like chromosome 8 here, and some regions, like chromosome 15, are more diverse. So these are the SNP density compared to the reference genome. So you can see that these are patterns where the ancestry switches at these changes in heterozygosity. Next, on this middle bar are the isolates that are most similar to this particular isolate, CH34, and we see that for parts of the genome, the Cook Turkish samples are most similar, but for this part here, the infantum is most similar. So this is why we hypothesized that one of the parents was similar to the Cook lineage for this particular sample, CH34, and one was similar to Leishmania infantum. And you can see further inside, this is nice genetic distance, and inside here is this ring of Somi. So we saw previously that CH34 and CH32 are quite different. And what you can see here in the same sort of plot for CH32 is its heterozygosity is lower, and for different portions of the genome. So if we switch back and forth, like this, you can see that the sections that are heterozygous between the two are different. So although these two isolates are genetically similar, their ancestries are not the exact same. And it's really important to note that we have this kind of patchy heterozygosity right across the genome, and it's different even if the isolates have a somewhat similar ancestry. And you can read more about this in the paper. The paper details that if we look at particular segments of the genome, we can compare these green samples from Cukurova in Turkey to the CH32 and CH34, and what we see is that some sections of the genome are very similar to Leishmania infantum from China. So you can see these strains like the dog strain and raccoon dog that are sampled from China, where we see segments of the DNA of the Cukurova and Cypriot strains fits inside a phylogeny drawn for representative samples. Not all segments of DNA have the same picture, but they appear to be very closely related to parts from China. So somehow they have segments of their genome that show very strong signals of common ancestry. So our other parent for these Cukurova and CH32 and CH34 is somewhat related to Leishmania infantum from China, or at least share common ancestry with them. And you see that further here, where we have a segment with is less closely related to those Leishmania infantum group 1 samples. And again here you see that there's more genetic distance, so it varies across the genome. Another feature of these parasites during the study is that the LD decay with distance here varies across groups. So if we look here, we have the LD estimate as the mean and standard deviation for 3 replicates for the different groups. This should decay with distance going from within a chromosomes down to between chromosomes. What you see for the LDON 2 here in purple is that those ones from the Nepalese uplands have very little LD decay, and that's because they're very, very genetically homogeneous as a group. In contrast, you see down here for the wine-coloured LDON 3 and pink-coloured LDON 5, they're from East Africa, the two. We see much higher levels of LD decay with distance and much more diversity. So the LD patterns within a particular sample collection is actually quite different. In terms of the indel diversity, what you see and fits in with other genomic work is that there's more insertions than deletions. So you see this right across the genome in general, with some exceptions. So indels are another feature of Leishmania genomic diversity that can be explored. In the same way, chromosome copy number changes varies extensively within each group, within samples, and over time. So what you can see in the standogram on the left is that the correlation of the SOBI signature across the different groups is not very similar. You see that the SNOMI is not a conserved feature, it changes rapidly, although most chromosomes shown in orange here are disomic, some are trisomic shown in green here, and some, like chromosome 31, tend to be tetrasomic. So it's worth considering that SOMI is a feature that can change very rapidly and really needs to be explored in a more homogeneous sample properly. Genomic data can give us information on CNVs, and what's really nice about this paper is that it gives us a derived an allele frequency spectrum for the genes that have CNVs. So you see that if we look down here at the spread column, that most genes have only a single isolate with dosage change. So that's the gene count is here going from 200, 400, 600. Most have a single isolate change, some have 2, fewer have 3, fewer again have 4, and so on and so on, and that decays so that when we go up to 100 genes, 100 of isolates with a CNV at this particular gene, that's very, very few. So this kind of fits what we'd expect for SNPs, and therefore means that we can use CNVs now for population genetic inference in terms of understanding the population structure, and allows us to see what CNVs are conserved across collections of samples. So if we draw phylogeny based on these CNVs, we see that CNVs tend to be quite variable. So some are ancestral, like the ones associated with this, the Indian subcontinent group here in red, or the infantum group, we see a small internal branch here, but many are not. So you see a lot of diversity in this collection of samples here. So look at CNVs with care. They may have signatures, they may not, and they are quite variable, just like SOMIs. SOMI changes, the same sort of pattern is present here. We do know that there are CNVs associated at drug-resistant genes that are frequent, may not be uniform. So here an example on chromosome 31, the miltefosine sensitivity locus of 4 genes. We see a deletion in many samples, shown here for the samples in blue, and we see this segment has no reads mapping to this region. So we see no coverage there, no read coverage at these 4 genes here shown in black. And we see certain samples that do have this, this segment like CH35 here. So genomic data can let us study CNVs at drug-resistant genes, and that's quite important for understanding molecular mechanisms of drug resistance. So as we draw to a close, we can think about what are the bigger questions in this field. So we've seen from the study that and other work that making better reference genomes is an ongoing area of work and is essential to understand the full genomic diversity in Leishmania parasites properly. There are ongoing questions over how sensibly do species mix and at what point in their life cycle do they do this most extensively. What does that mean if we have sandflies that are feeding multiple times? Related to this is how much recombination occurs across chromosomes and how often. Similarly, if you think about drug resistance, it's aspects of it are sometimes polygenic in these parasites, so it can be developed, for instance, polygenic risk scores to give us information on predicting drug resistance based on genomic data alone. So the last point here should say, what about the vectors? So the vector biology is not well understood, really, it's not well researched, and we need to study Leishmania inside the vectors to understand what changes they're undergoing in their life cycle, how they recombine, and how they mix. And then finally, what happens inside the patient in terms of post-calorizar dermal leishmaniasis. Why does relapse happen? What's happened to the parasite when they're inside the patient so many times? What happens if people get reinfected? There are many, many questions that we really have not tackled in this area. So there's a large number of people I really must acknowledge, including most prominently Suzanne Franson and other people at the Sanger Institute, including James Cotton, Mandy Sanders, Matt Berman, Matt Rogers, and Caroline Durant. At the Institute of Tropical Medicine in Antwerp, I'd like to also acknowledge the work done on this by Jean-Claude Dujardin, Hideo Imamura, and Frederick Vandenberg. At Charité University in Berlin, I'd like to acknowledge work by Olivia Stark, Bettina Moser, Gabriele Schunian, who really contributed a lot. So what I have here next is some suggested reading to give you background in this area, including information in the epidemiology, the phylogenetics of Leishmania in terms of their especially the history, other by Cools and Mauricio on this area too, Imamura and Dujardin's guide to genomics of Leishmania, the paper by France, Suzanne et al., a recent paper on intracellular Leishmania parasite genomes directly sequenced from patients, which is quite important for current research. Rougeron's primer on Leishmania PopGen, and also the work on genome and transcription analysis of Leishmania. So finally, I'd like to suggest that you explore microreact further to get a feel for the geographic, phylogenetic, and genomic diversity of the sample collection, and hopefully you can use microreact for your own projects and really allow other people to interact with your work. And as I said before, if we go to the start slide, If you have questions, just give me a shout on Twitter. I'll be happy to answer your questions. I think this is a really great platform for communicating with scientists, for creating a community, and I'm happy to answer anything further. Thank you for your time, and also thanks to Chris again for organising an excellent workshop.